Hi, my name is Amir Tataibi, an assistant professor of biomedical engineering, and in this series we talk about physiological systems in biomechanics. In the previous video we talked about neurons, the fundamental units of our nervous system. Also we talked about a synapse, which is a structure that permits the transfer of electrical signals from a previous nerve cell to a next nerve cell, from a presynaptic membrane to a postsynaptic membrane. So if you haven't watched that video, I encourage you to watch it first and then return to this video. In this video, we want to talk about a synapse and how an electrical current is transferred from a presynaptic membrane to a postsynaptic membrane in the next nerve cell. So first, let's start with some definitions. Here we have a presynaptic membrane and a postsynaptic membrane. The gap between the presynaptic membrane and the postsynaptic membrane is called a synaptic cleft. On the presynaptic membrane, there are some voltage-gated calcium channels. There are also ligand-gated ion channels on the surface of the postsynaptic membrane. Other components exist, such as mitochondrion, as well as synaptic vesicles, which include neurotransmitters. We will talk about these neurotransmitters in more detail. So, as a result of action potentials in the presynaptic axon, an electric current reaches the axon terminal, the presynaptic axon terminal. This electric current changes the voltage in the intracellular space, and as a result, opens the voltage-gated calcium channels. Therefore, the calcium ions move into the presynaptic cell. And then they activate some enzymes that turn on small motors that pull the vesicles, the synaptic vesicles, to the synaptic cleft and fuse it with the synaptic membrane. As a result, the neurotransmitters inside the synaptic vesicles are released into the gap between the presynaptic membrane and the postsynaptic membrane, the synaptic cleft. These neurotransmitters, these are some ligands that they can open the ion channels located on the postsynaptic membrane. So they are released into the synaptic cleft, then they attach to the attachment sites on the ligand-gated ion channels in the postsynaptic membrane, open those channels, and as a result, electric signals can enter the postsynaptic membrane. The neurotransmitters can be excitatory or inhibitory neurotransmitters. The excitatory neurotransmitters, they usually open sodium channels. As a result, positive sodium ions move from the extracellular space to the intracellular space of the postsynaptic membrane. The inhibitory neurotransmitters, on the other side, they open the potassium channels or the chloride channels. As a result of opening the potassium channels, for example, the potassium ions move from the intracellular space of the postsynaptic cell to the extracellular space. Therefore, we will have a negative flow of ions because the positive charges are moving outside of the cell, a negative flow into the cell, let's say. That's why they are called inhibitory neurotransmitters. Through this process, the electrical signal passes from the presynaptic membrane to the postsynaptic membrane. Thank you for watching this video. Until the next one, bye.